So now what we're going to do is switch gears a little bit and compare what we've just seen with Saccharomyces to a different fungus. Now this other fungus is called Mucilagomatus, and we'll talk in greater detail about that, actually give you some images that are better than the ones that I'm drawing a little bit later. But this is a fungus that is a plant pathogen. And we'll talk about that when you can see better images. And the other thing about it that's interesting is it served as an important genetic model system by two people that have done stuff we've already heard about. So it's a model system, and one of the people who worked on it was Robin Holliday, that we talked about for the Holliday model for recombination. And a current really big investigator that uses Mucilagomatus is Regina Common, who is currently who has been the head of a Max Planck Institute in Marburg, Germany, that focused on basically this organism. There are probably over 50 investigators that were working exclusively on this organism. And she's currently, I think, still acting director while they look for a new director. She was, I don't know how many years she's been director of the Max Planck. But anyway, the reason that I bring her up is before she got into working with Mucilagomatus, she worked with mu and the gin protein. If you recall from when we were talking about transposition and mu and site-specific recombination, she is the person that worked on and figured out a lot of stuff related to that before she got into working with Mucilagomatus. Okay. Now Mucilagomatus, just as Saccharomyces, has haploid cells. So these are haploid cells of Mucilagomatus, and they're yeast-like, although as you can see, they look more cigar-shaped than the more ovoid cells of yeast, and they reproduce by budding. And just as we saw with Saccharomyces, they come in two flavors, but that's really an oversimplification. So if you have a mating type cell that is A1, it's haploid. And if you have another cell which is A2, it's also haploid. And just as we saw for Saccharomyces, these different cell types can produce pheromones that they secrete. So the A1s will secrete A1 pheromone. And the A2 cells have a receptor for that particular pheromone. And similarly, the A2 cells secrete their pheromone, which can then be received by a receptor that's present on the cell of the A1 cell. Okay? And then the result of this, which is different than in Saccharomyces, is when these two cell types communicate like this, they do come together and fuse. But the result is not a diploid cell. Instead, what happens is they produce what's called a dikaryon. So instead of reproducing by budding, they produce this filamentous structure that will have the nucleus for the A1 separate from the nucleus for the A2. And what's important about this filamentous structure is now it becomes infectious. And it can penetrate the host plant. Okay. So now you have a difference between Saccharomyces and Mucilago in that this mating process is required for the fungus to carry out its life cycle, which is obligately parasitic, meaning that it has to go through this process of infecting a plant to complete its sexual life cycle. Okay? Now, the loci that are responsible for controlling this are actually two different loci. So this is a difference from Saccharomyces as well. There's the A locus, and then there's a separate locus called the B locus. So the A locus, there are only two types, A1 or A2, which we've illustrated over there. And just as we saw with Saccharomyces, they're not allelic. So the A1 encodes the A1 pheromone and the receptor for the other pheromone. The A2 codes for the A2 pheromone and the receptor for the A1 pheromone. And also it turns out that in the A2, there are genes that are involved in controlling mitochondrial inheritance when this mating process occurs. So when these two cell types fuse and produce an infectious dikaryon, only one of the mitochondrial types gets passed on to the next generation. That's probably an oversimplification, but there's strong evidence that at least predominantly the A2 is able to control inheritance of its mitochondria and get rid of the other kind of mitochondria. And that's in the A locus. Now, again, if you look at the structure of those loci, they're not homologous to each other. And so we don't really call them allelic, but instead we refer to them as the A1 idiomorph and the A2 idiomorph. Okay? Now, there's a second locus, though, that's a part of this process. And this second locus, the B locus, determines whether or not you actually get infectious dikaryon produced. And so the way I would represent this, it's kind of like, I don't know how many of you guys get involved in these sort of dating sites, you know, like eHarmony or Match.com or, I don't know, Tinder, whatever you guys get involved in. So I kind of liken this to those kinds of matching possibilities. So it's kind of like the A locus is kind of like, say on Match.com, you find somebody who is interested in reading, you know, likes books, okay? And you have an interest in books. And so you decide, okay, what we're gonna do is we're not gonna have, this is gonna be low pressure, we're gonna get together, have coffee maybe, or, you know, maybe a drink, but probably coffee. And we're gonna see how things work out. So that's where the second part comes in. It's kind of like, that's the first date, if you want to call it a date, but the first get together is, okay, we both like books. You get together and the B locus says, okay, well, you have that in common. Are things should we proceed? And what you find out at that particular meeting is that while you like Dostoevsky and Tolstoy, that's the literature you like to read, this potential mate that you've gotten together with is more into graphic novels and, you know, Marvel comic books or whatever. And you go, nah, I don't really think that's a good match. And so the second meeting was the next gatekeeper of whether we're going to go on further. And this B locus, it turns out that those different forms are alleles, and they've identified 20 to 25 different forms of B locus. And it's critical that those two forms be different in the mating partners. So just as you had to be different between A1 and A2 in order to get that initial, we're going to come together and and have this meeting. After that's happened, then at the B locus you have to be different as well. So in this case, it's not so much that you have the same interest in reading, it's that they have to be different in order for it to be successful. So somebody had this idea in their Match.com algorithm that if you like the same stuff, that's not going to work. And that's kind of the two different ways in which the mating is controlled. Now, this B locus, it encodes transcription factors, and specifically it encodes what we referred to earlier in the semester, but since exam 1, homeodomain proteins. So homeodomain proteins, anybody remember what the homeodomain DNA binding domain is used for typically?
It's like a differentiation.
Yeah, so for development. So homeodomains are used in controlling developmental genes. Okay, and so this is, there are actually two proteins that are encoded in the B locus. You get the B east, and you also get the B west. And both of those have to be different in the mating partners relative to their potential mate if this is going to be successful. So what happens is from this single locus, you get two proteins produced, B east and B west. And if mating is going to be successful, and we're going to go on to produce a filamentous dikaryon, then the B east from one of the partners has to be different than the B east that the other partner has. And if so, it can come together with the B west from the other partner. So you produce a heterodimer that becomes an active transcription factor for the pathogenic program. And without that, you don't get successful production of the dikaryon, and the fungus is not able to infect the plant. Okay, and just one other piece of information is that again you've got A1 and A2 pheromones, and they are initially encoded by genes and then processed down to the small peptides, kind of like what we saw in Saccharomyces, and they also get prenylation, which means adding a lipid, and carboxymethylation. So they get modified similarly to what we saw in yeast. Okay, so before we move on, just one question in terms of what you've seen with Mucilago and what you saw with Saccharomyces. So how are the mating loci, and in particular the A locus in Mucilago, similar or different from the MAT-A, MAT-alpha in Saccharomyces? So what's encoded by the A locus in Mucilago?
Pheromones and receptors.
Yeah, pheromones and receptors. What's encoded by the MAT-A or MAT-alpha locus in Saccharomyces? Are they?
But also the...
Does the pheromone or receptor get encoded by the MAT locus in Saccharomyces? No.
No.
Okay. And so as we saw a while back, the MAT loci and Saccharomyces encode either transcriptional activators or repressors, right? And those regulate expression of pheromone and receptor genes and other genes.
Okay.
So if you recall, in the yeast situation, you had these proteins that either block or help transcription of all the haploid-specific genes, whereas in Mucilago, the A1 and A2, they directly code for pheromone and receptor. So this is directly encoded in the A mating type locus, versus in Saccharomyces, where they encode regulatory transcription factors instead. Okay, so that's another take-home lesson in terms of how they do it differently. Okay, and with that, we'll take a short break before we move on to the next set of discussion.